So lipids, structure and function part 2. Here we're going to cover steroids and phospholipids. But let's refresh our memory really quickly. Main types of lipids, fats, which we covered in a previous video. Their main function is storage of energy. Here we're going to cover phospholipids, which are the main component of cell membranes, and steroids, which one of their main functions is cell signaling. And again, these three molecules all have very different structure, so why are we linking them together? Why are we grouping them together in this one category of lipids? Because they're all very hydrophobic. Because most of the bonds that are in these molecules are carbon-carbon or carbon-hydrogen bonds, which are very nonpolar, which means they don't have any slight charges to the molecule, so therefore water cannot make hydrogen bonds with them, right? So these again are general structures of the three different types of molecules. Here we have a triglyceride, here we have a phospholipid, here we have a steroid. You should be able to recognize the structure of each of these molecules. So if I show you an image like this, you should be like, okay, that's a steroid, then the main function of steroids is this. Phospholipids. Phospholipids are one of the most important molecules as far as in the development of life, in the evolution of life on our planet. They are a variation on a triglyceride. So they're a variation on triglycerides. So they have this glycerol molecule right here, and we see two of those three carbons are bonded in the glycerol are bonded to fatty acid chains. But instead of having a third fatty acid chain, they have a phosphate group. Okay, so what do we know about the characteristics of this molecule, especially in terms of how it would interact with water? Right, so we have these fatty acid chains are very nonpolar, therefore they're very hydrophobic. This region of the molecule, though, with this phosphate group, that oxygen is a negative charge, phosphate, oxygen, all of these bonds in here are very polar. This region of the phospholipid molecule will be very hydrophilic because it is very polar. So this gives the phospholipids a very unique role in cells because they have this dual nature of being both hydrophilic and hydrophobic. Usually when we're simplifying phospholipids, we'll draw them like this little like circle with two tails kind of coming off of it. That's our simplified version, our simplified drawing of a phospholipid molecule. So we'll see this a lot as we continue to look at phospholipids, especially once we start talking about cell membranes. Fatty acid chains coming off the phospholipids may or may not be saturated or unsaturated. So usually in most cells, one of these fatty acid chains will be unsaturated. It'll have at least one or more double bonds in that fatty acid chain. But why do we care? Why are they important? So again, that dual nature of having both a hydrophobic region and a hydrophilic region gives these molecules the unique ability to both interact with water as well as interact with hydrophobic substances or other lipids, right? And so what happens, if we were to take a bunch of these molecules, just a pure sample of phospholipids and drop them into water, they would spontaneously arrange themselves into either a micelle, which is this right here, or into a phospholipid bilayer, okay? And the reason why they do that is because these polar heads are attracted to the water, they're hydrophilic, they can make hydrogen bonds with water, and these tails, these fatty acid tails, they're hydrophobic. So they're going to have hydrophobic interactions attracting each other. Okay, those tails are going to be attracted to each other, and the phosphate heads are going to be facing out towards water. And this is what makes up basically the cell membrane, most of the cell membrane. The double layer of phospholipid tails, these hydrophobic fatty acid tails of the phospholipid molecule. And then outside the cell, let's say maybe this was the outside of the cell, cells are usually in an aqueous environment. So these phospholipid heads, these phosphate groups of the phospholipid molecule would be hydrophilic and readily interact and make bonds with water. Hydrophobic molecules attract each other and they kind of clump together when in the presence of water. So these hydrophobic tails, these fatty acid tails of the phospholipid molecule, are going to be attracted to each other and make that either that double layer in the sense of the cell membrane, or sometimes they make this what we call a micelle, where the hydrophobic tails just kind of all face in together and we get basically a ball of the phospholipid phosphate heads facing out. And we'll dive in more into the structure of the cell membrane in the future in another video. So our last class of lipids that I wanted to cover for us are the steroids. Okay, we're just going to talk generally about steroids, their structure, and their general function in living organisms. So most steroids have a basic structure like this, where they have 1, 2, 3 six-sided rings of carbon-hydrogen, and then one 5-sided ring. Okay, so again, remember when we're looking at all of these molecules like this, any point represents a carbon-carbon bond, and then we can assume there's associated hydrogen bonds to fill in carbon's 4 total bonds. Any other elements that are part of this molecule that are not carbon or hydrogen are typically indicated. So like this oxygen right here, this would be the only oxygen in this entire molecule. So this is cholesterol. Most steroids are built by some sort of alteration of cholesterol, right? So the cholesterol molecule. But regardless, they're still very hydrophobic, right? All those carbon-carbon, carbon-hydrogen bonds, very nonpolar, so therefore this is a very hydrophobic molecule. But how we get to different types of steroids, because you've probably heard of different types of steroids, testosterone, estrogen, progesterone, right? We take this basic molecule and we alter its functional groups. So maybe we take off this long chain of carbon hydrogen right here and we put something else there. Or maybe we break up this double bond and put something else here. Okay, so we can alter different functional groups all over this molecule, but we retain that same basic 4 rings of carbon structure, which is steroids. So an example would be aldosterone and cortisol. Okay, so this is cortisol right here, this is aldosterone. These are both hormones that are produced in our stress response in humans. So aldosterone, if we look at it here, its functional groups are slightly different, but we still have 1, 2, 3, 4 rings of carbon, 1, 2, 3, 4 rings of carbon in both of these. Okay, so that base structure is still the same. But if we notice, there are some very subtle differences, right? So right here, coming off right here, we have an oxygen, okay? And right here, there's no oxygen, right? There's no double bond to that oxygen, so we've altered that functional group. Same thing with this portion of the molecule. Right, we have an extra hydroxyl group right here, and right here, we don't have that extra hydroxyl group, yeah? So we just altered these few slight changes mean that these hormones, these steroids, are going to have very different effects on the cells that they come into contact with. And different cells will react to these hormones differently. So for example, aldosterone, one of its functions is in the kidneys. It helps to, with the reabsorption of sodium ions, which influences water absorption, right? Remember, if we think about how water moves in osmosis, it helps to up our blood pressure, right? Which if we're in a stressful environment, that could potentially be helpful if we're about to like flee from something or about to run away from something that's going to eat us. Higher blood pressure means that we can move more blood around. Cortisol, even though it's very similar, has slightly different functions and it's going to affect a totally different set of cells. So cortisol helps to increase blood glucose levels, so it starts to help to break down glycogen in our liver and release that glucose that was stored in glycogen from our liver into our blood. It also helps to suppress the immune response, so it's going to impact our immune cells and suppress their activity. And it also helps to ramp up the metabolism of converting glucose into ATP in our a lot of times our muscle cells, but other cells of our body as well, right? But both of these are really, their intention is to help to get us ready to fight or fly from something, right? It's either to fight something or to run away from it, and that's their intention. But again, the key piece here, they're still lipids, even though they have a totally different structure than phospholipids or triglycerides, because they're mostly carbon-carbon or carbon-hydrogen bonds. And then we just alter these various functional groups, and then that gives these steroids very different functions.